High-throughput screening combined with mass spectrometry is a next-generation screening technology that can screen even challenging targets. The conventional biochemical methods using fluorescence, luminescence, or radioactive substances often cause false positive and false negative issues attributed to each of the technologies. In contrast, mass spectrometry allows direct and label-free detection of multiple target molecules with high sensitivity sufficient to monitor small mass differences among analytes in assay system. This characteristic of mass spectrometry-based approach provides high accuracy and reproducibility for the identification of hit compounds in screening campaigns. Axcelead has developed high-throughput-mass spectrometry platforms, HTMS, by installing the RapidFire system onto mass spectrometer. Our HTMS takes only 10 seconds to acquire single data point and consequently achieved a more than 10 times faster readout than LC-MS methods. As a result, this system accelerates screening campaigns and enables us to identify hit compounds from extensive library in a short period of time. Furthermore, we have a lot of experience and confidence in handling mass spectrometry-based screening processes. Axcelead has applied our HTMS technology for approximately 35 projects since 2009 and successfully identified hit compounds in almost all the projects. Axcelead has a highly experienced team in high-throughput screening using mass spectrometry, and we are always ready to meet your diverse needs. Please let us know if you are interested in our HTMS platform. We would be delighted to hear from you.